Now we are starting embryology series, and first we will discuss about human development phase from days 1 to days 4. So in human development, step 1 is fertilization. Fertilization is the first step in human development. During fertilization process, sperm meets with egg. Sperm mates with egg and it forms zygote. Zygote then eventually differentiate into baby. When semen enter into the vagina, basically it neutralizes it acidic environment of the vagina, and most of the sperm are dead because they stuck in vaginal mucus, because there is a lot of mucus in the vagina, and they mostly lost in cervix and some observed by white blood cells during their journey to the fallopian tube. Only few survivors left. Few survivor sperms. They are almost 1,000. They are survivors. Survivors sperm left out of millions. So what they do? They rub against wall. So this wall, they rub against this wall. The sperm rub against this wall and remove glycoprotein coat. This process is called capacitation. Capacitation is process in which sperm rub against wall of the uterus, wall of the ovary and remove its glycoprotein coat, and sperm also release, the acrosome of the sperm also release some enzyme. These enzymes are called hyaluronidase. These hyaluronidase, they break down hyaluronic acid. Hyaluronic acid, it protects egg. So basically they break down this protective coat of the egg. Egg is the largest cell in the human body, and sperm is very small as compared to the egg. It is almost 1/30th size of the egg, and it is very thin, and egg is very largest cell. Egg has some layers. We will name it corona radiata. It is outer layer of the egg. Zona pellucida or sperm binding protein 3, CP3. This is other layer of the egg cell, and when zona pellucida, it is jelly like. Zona pellucida, it is jelly like layer of the egg. It is extracellular matrix of glycoprotein. Zona pellucida is made up of glycoprotein, and corona radiata, these blue lines, these are corona radiata, these are outer coverings of the egg. The corona radiata, these are follicular cells. When some sperm, these travel from corona radiata and it connect with zona pellucida. It actin anchors sperm, and acrosomal reaction takes place. Sperm release acrosine and sperm and egg fuse. It is called sperm binding. Sperm fuse with this layer of zona pellucida. Inside the egg cell contain a lot of cortical granules. These are cortical granules. These are located inside the cell. So these cortical granules. These are basically bags of enzyme like peroxidases. So when sperm connect with the egg membrane, so calcium level rises, and after calcium level rises, then cortical reaction takes place and granules fuse with cell membrane. These cortical granules fuse with one another and with calcium level and water level, and they make hyaline layer with the space of egg, and they only allow only one sperm to enter inside the egg, and they prevent other sperms cannot enter into the cell because of this hyaline layer. Because of it protects from polyspermia and more sperm fusing with the egg. So this hyaline layer is very important. After that, only a single sperm out of millions of sperms enter into the egg cells. It is like a marathon or like winning a lottery out of millions. Only single sperm enter inside the egg cell. During this phase, meiosis II takes place inside the egg cell, and it releases one second polar body. Second polar body release, and female pronucleus is present. Pronucleus, and second polar body is apoptosis takes place, and second polar body disappears, and only female pronucleus is now remaining. This is female pronucleus inside the egg. Only one sperm that enters into the cell, it comes close to the female pronucleus, and it shed its sugars, and it also become these two, it becomes pronucleus. This is female pronucleus, and this is male pronucleus. So they come closer and they exchange genetic material with one another. At this stage, female pronucleus and male pronucleus, they exchange genetic information and they form complementary pair, and eventually they form zygote. Zygote is basically diploid. So they exchange genetic information with one another and form zygote. So now I will recap this whole process. Sperm enter into the vagina. Then it comes into the cervix. Then it goes into the uterus. Then from uterine tube, in the uterine tube is also known as fallopian tube. It enter into the egg. By crossing layers of egg like corona radiata, it is layer of the egg, corona radiata, zona pellucida, and it exchange genetic material. Genetic material is exchanged, and it form eventually a big cell called zygote.